Aloha. Today I will begin with Chapter 23, discussing the hormonal regulation and integration of the mammalian metabolism. This chapter is unique in the way it presents its material, and but mostly because you will learn very little new facts in this chapter. In the previous chapters, the pathways were new for you, the enzyme names, the metabolites was pretty much all new to you. But in this chapter, we will discuss regulatory mechanisms and functionalities that we have already talked about. It's just that we will unite them together. Instead of looking at individual pathways, we will look at the whole metabolism as a whole, and hopefully that will give you a better appreciation of what you have learned and how that applies to the function of various tissues that rely heavily on metabolism. Not that there are tissues that don't rely on metabolism, obviously they all do, but there are some that have more critical role in the metabolism as a whole. So I begin this presentation usually with a conversation of an exchange between me and you. I ask questions and you provide answers, and these are questions that are not very difficult, and for the most part you know how to answer them, and we just have just a natural conversation that would start between us. Obviously this is not going to happen this time, so I might be asking a lot of rhetorical questions that I would answer myself, but hopefully you will appreciate the logic of the questions and answers, and leading you towards gaining a better understanding how the regulation of our metabolism in general, in very general principles, is actually happening. Alright, so since we're talking about hormonal regulation, it is natural to start the discussion or the presentation, since it's not really a discussion, with a question: what are hormones? And you probably already know, but these are molecules that are, well, some of them are smaller, some of them are larger, but they're endocrine molecules that are synthesized by the endocrine system. And what makes a system being called endocrine? Well, something typical for the endocrine system is that they synthesize bioactive molecules that are released in the blood, and then through the blood is disseminated throughout the entire body. That is something characteristic and defining for the endocrine system. So in the sense, these hormones can reach targets that are pretty much anywhere that the blood can reach. So as signaling molecules, what is the role of the hormones? Well, hormones regulate, and at the same time they integrate or unite all processes together, because one hormone can regulate or control multiple processes, which is very characteristic for hormones. So their role is to control and to integrate. What processes do you know to be hormone regulated? Virtually all. I cannot think of, off the top of my head, of any pathway that is not somehow or any function that is not somehow regulated by hormones. Someone might say, well, perhaps taste or smell, well, or vision for that matter. Even these processes at some point will be regulated by a hormonal function. Even if it's just expression, that's still hormonal regulation. What regulates the hormones? What regulates the regulators? Hormones are generally regulated by something called a hormonal hierarchy, but ultimately they're controlled by the central nervous system. So how does the central nervous system know what and how to regulate? Well, that is because the central nervous system uses information that is received by various sensors and receptors throughout our body. The receptors perceived changes in their environment, and they send signals to the central nervous system that then tells the hormones how to behave and how to react, whether to be released or to be retained within the cell. Are there other molecules other than hormones with regulatory functions? Of course there are. There are many different bioactive molecules. Some of them are well known are the neurotransmitters or other bioactive molecules, which based on their target can either act as autocrine signals, paracrine signals, and endocrine signals. We've talked about those in 481. Autocrine when they act on their own cells, paracrine when they act on the neighboring cells, and endocrine is when they are secreted in the blood and go everywhere in the body. Now, hormones, neurotransmitters, and all bioactive molecules, they are part of the so-called neuroendocrine system, which controls, coordinates, and unites all metabolic processes in the body. So how does a hormone deliver its message to a cell? And why different cells have different responses to the same hormone? It's the same hormone. How can it exert different responses? And why would the cell react when a hormone is secreted? And how is the message which the hormone carries be transferred into the cell? You're all familiar with that because you know biosignaling, and that is that the cell's response depend on the type of receptors and downstream effector molecules activated by a hormone. It doesn't matter what hormone binds. Once bound to a receptor, it matters what receptor that receptor is bound to and what further downstream molecules will be activated. That will determine what is the effect. Sometimes, as you know, we've talked about how sometimes insulin receptor hijacks the beta G-protein coupled receptor signaling. We discussed this in 481. You can somehow activate downstream molecules of other receptors that normally don't signal with the receptor that is being activated. Regardless of whether that's the receptor or not, if something binds that receptor, the receptor will exert its function. So, without going any deeper than this, the reason why cells know or cells react on the release of hormones is because they have receptor that can bind that hormone. If there is no receptors for insulin, or the insulin receptors are no longer sensitive for a certain concentration of insulin, the person will develop diabetes. Okay, let's move on, because we can talk about this for a long time. Where are the receptors localized? They're usually on membranes, cell membrane or nuclear membranes. Now, the concentration of hormones in the blood is usually low. So how can hormones then exert such powerful effect? And what does that tell you about the hormone receptor interaction? Now, you know that the characteristics of the signaling process are that every signal and ligand interaction is characterized by affinity and specificity. And again, affinity describes the strength of the interaction, and specificity describes the selectivity of that interaction. So, the hormone receptor interaction is usually with high affinity. I mean, everything is relative, of course, with high affinity and high specificity. But why such small concentration? I mean, even if such small concentration will bind those receptors, that means that the interaction is strong. That's why the receptor has high affinity for the hormone and high selectivity as well. But why then there will be such powerful effect? Remember, the other characteristics of the signaling process was amplification. What happens is the so-called downstream enzymatic cascades. A single receptor can activate a few proteins directly, but then these few proteins, each of them will activate a few of their own targets, and each of those few targets will activate a few targets, and as you can see how this increases dramatically, and then you may have one molecule binding one receptor, but you have a massive response from the cell or multiple cells for that matter. So through amplification, a single signal can become a massive response. So some responses are fast and some slow. Why is that? Well, fast responses are usually connected with response elements such as channels or enzymes that usually exert fast response. The response is connected with their function, and all it takes is for them to be either turned on or turned off. Now, slow responses are usually connected or dependent on gene regulation. It takes time for the signal to reach the nucleus, to stimulate all the processes that are governing the expression of a gene. Then it takes time for mRNA to be synthesized, to be exported from the nucleus, and then to be translated, for that protein to be processed, and perhaps even get to the surface of the cell. That all takes time. It could be hours, it could be days, and sometimes it takes even longer if a certain level of desired response is needed. So that's why we have fast, which is usually for channel. We turn on the channel, the concentration gradient changes. We open the channel, or we turn on an enzyme, and enzyme starts doing its function. There's no need, there's no time to wait. But if we need to express more enzymes, then that will take a little longer, not just an instant reply or response. So which hormones will have fast and which slow response? Cell or nuclear membrane receptor hormones? And why is that? The cell membrane receptors, or hormones that bind cell membrane receptors, usually elicit a fast response, while the nuclear membrane, the hormones that bind to the receptors located on the nuclear membrane, usually exert slow response. Cell membrane receptor signaling usually leads to fast response because they are connected, as I've mentioned, with membrane reactive elements such as channels or transporters and cytosolic enzymes. While the nuclear membrane receptors usually signal by controlling gene regulation, and like I said, that is why the response is slower. So the fact that we need process integration tells you what about the tissues in our body? Well, the fact that you need to unite things together, that means that they're different. If something needs to be united with something else, that means that they're individual entities, and they perhaps have specialized functions. Even in our societies this way, if we need to unite certain groups of people, that means that they're already separate. If they were together, we wouldn't need to unite them. All of our tissues have specialized function, but they work as a whole. Something needs to control all that. Something needs to bring them all together so that they can work in unison. If they're not working together, then there's chaos and there will be no progress. So there has to be a balance between all independent functions. None of them is more important than the other in principle. Now, some of them might have more critical role than others, but that doesn't mean that others are not important. All right, so this is depicted in this image by showing how different tissues have certain functions, but they all work together. Just like the liver cannot function without the blood that is pumped by the heart also will not be able to function if the liver does not produce the necessary nutrients to supply energy to the heart. Okay, so let's now look at the more specifically what happens in the different tissues. And we're going to look into the hepatocyte, what happens in the hepatocytes, and the myocytes, and adipocytes, and at the end, the neurons. All right, and like I said, you're probably not going to see much new information here, but hopefully it will help you appreciate what you've learned so far. All right, so hepatocytes, or the liver cells, they are, I've learned this, by the way, in 4th grade. I remember in my biology class, I vividly remember how I read in my biology textbook that the chemical laboratory of the body is the liver. And indeed, that is the case. The liver processes everything. The liver supplies all other tissues with nutrients and a proper environment. So it's natural for us to look into what happens in terms of the major metabolic pathways, carbohydrates, lipids, and proteins. And like I said, you already know that. But once glucose is in the cell, it gets immediately phosphorylated into glucose 6-phosphate. This way it doesn't come out. And once glucose phosphate is inside, then based on what the needs of the body and the liver are, that will determine what's going to happen with glucose 6-phosphate. If there is a need for energy, it will be degraded to glycolysis to pyruvate, which will then become acetyl-CoA. And if again acetyl-CoA is needed for energy, it will enter the citric acid cycle and be degraded to carbon dioxide. All released electrons then will be used in the oxidative phosphorylation pathway to make ATP and water from oxygen. Now, if we have plenty of energy or specific needs, that acetyl-CoA also can be used for fatty acid synthesis that can be stored, or for cholesterol synthesis for whatever reason that is. The cholesterol is going to be used. Now, if synthetic processes are also in need, if you have plenty of energy, glucose 6-phosphate will also enter the pentose phosphate pathway that will produce the desired NADPH needed for biosynthetic processes such as fatty acid synthesis and nucleotide synthesis. Now, if that glucose 6-phosphate was a product of glycogen degradation, then obviously the glucose 6-phosphate will be exported into the blood to supply the entire organism with the necessary glucose. And I should have mentioned this before when we had glucose in excess, that some of that glucose will be stored as liver glycogen. Now, what happens with the amino acids? Well, proteins get degraded into amino acids. Amino acids will be delivered from the intestines to the liver, and then again, based on what they're needed for, they will have a different fate. And most commonly, the amino acids are going to be used as building blocks for various proteins, whether by the liver or whether by extrahepatic tissues. Whatever it is, they will be used for biosynthetic purposes. Now, if there is a need of energy and there's no other energy source, primarily glucose, some of the amino acids, or many of them, will be degraded. In the process of their degradation, ammonia will be generated that later on will be excreted in the form of urea. And then the resulting products of the amino acids degradation, and there are various products, then can be used for whatever purposes they can be used. They can all be burned in the citric acid cycle into carbon dioxide and electrons that will be used for ATP synthesis. Or, as is shown here, the various products of the amino acid degradation can be used for glucose synthesis, or for ketone body synthesis. I'm trying to find out where is that shown on this image, but it's not shown. But as we know, there are two types of amino acid: glucogenic, those that generate glucose. It shows here glycogen, but that's very uncommon. Or ketone bodies, if during starvation. Now, if we take too much proteins and too much carbs, obviously, and we don't need a lot of proteins to synthesize, a lot of amino acids to synthesize proteins, then the generated acetyl-CoA can be used for fatty acid synthesis and the storage of lipids. What happens with lipids in the liver? Well, once the fatty acids enter the liver cells, they either get packaged and sent out, right here, for the other tissues to use for energy, or the liver uses them for energy, shown here by degradation to acetyl-CoA, and then citric acid cycle and ATP production, or they're stored as liver lipids. These are just the liver cells have that ability to store certain amount of fat in its own cells. Now, the more that happens, the worse are the consequences for the liver. As a matter of fact, people that abuse alcohol or abuse fat, and that is normally not used for energy as a primary energy source, they develop something known as fatty liver, where a lot of fats are deposited in the liver. In addition to that, when acetyl-CoA is generated from the fatty acid degradation, if that happens during starvation, that will be used for ketone bodies production, and that will be exported. That will leave the cells and then be used by all other tissues. Acetyl-CoA also can be used for the generation of cholesterol, and obviously later on, cholesterol is used for bile salts, steroid hormones, and vitamin D production. Now notice that there's no fatty acid production. That is because, as you already know, we don't degrade fat to generate fat. Or, to be more precise, we don't degrade fatty acids to generate fatty acids. That's just going to cause production of heat, and that'll be it. Alright, so this is all about lipids in general. Now, imagine we don't have one cell that does lipids, another one that does proteins, another one that does glucose, and then everything else, by the way, that happens in the hepatic cell. They're all happening at the same time. Well, maybe not absolutely everything, but they all work in unison together. If there is starvation, the lipid, protein, and carbohydrate metabolism adjust accordingly so that they can work together. If there is plenty of energy, they adjust themselves so that they can work together to supply the necessary energy and resources for the cell and the organism as a whole. That means that there must be a tight regulation. And we have a good understanding of what goes on, but we're far from knowing everything and all the details related to the metabolic regulation. Alright, well, enough with hepatocytes. Now let's talk more about adipocytes. These are another name for, or the official name of fat cells. We have Well, there's multiple types of fat, but in general, there's two types, white and brown fat. And their primary function is lipids storage and supply. Now, wide lipid cells, or white adipocytes, as you can see them, they are larger, they're more round, with a single large lipid droplet. They have fully active metabolism in them and ATP synthesis, and their primary function is to deposit triglycerides. That means that they can synthesize them, but they can also break them apart into fatty acids. And then the fatty acids are excreted in the blood and then delivered to the tissues that need them. The brown adipocytes, though, as you can see here in this diagram, they are smaller. They have a lot more mitochondria, and their droplets, well, they have multiple droplets, not just a single large droplet. Now, something interesting that they also contain is that they are rich on the thermogenin protein. It's also known as an uncoupling protein, and that is because it allows the entrance of protons through it and skipping, and for these protons to skip entering through the ATP synthase. By doing so, the thermogenin, or the uncoupling protein, allows for the protons to come in, and in the process it generates heat. So that's why the brown adipocytes, they usually exist in infants and, well, newborns. And the older the child gets, the less brown fat they have. And one of the primary function of the brown fat is to generate heat, and that heat is generated by allowing the protons to pass through the thermogenin, and the heat keeps major vessels and major organs in more of a constant temperature in small bodies such as newborns. As you know, they're so small that the hypothermia can easily affect them. And I guess it's just a preservation mechanism to keep the heat and energy within the body, especially around these major organs. Now, adults pretty much have no brown fat left because I guess we have plenty of the other one to keep us warm. I'm joking, of course, but the need of, our bodies become larger, and the need of that strict control is much lower, and so that's why we, the adults, almost have no brown fat. Okay, the next cells or tissue we should talk about are the myocytes. Primary function, make or use ATP. There are two types of myocytes or muscles, red muscles and white muscles. The red are known as slow twitch. Slow twitch muscles are adapted for slower, prolonged work. And they tire slowly. In other words, you can use them for a long time. They require a higher and more constant and reliable supply of blood. That's why they have a rich capillary system. The red muscle tissue has a rich capillary system, and they that also allow them for ready access to ATP and steady production of ATP. The fast twitch, or the white muscles, are kind of opposite to the red muscles. They're adapted for fast work, but they're more prone to fatigue. And they use a lot of ATP, but they cannot replace it fast enough. They also have poor blood supply, and for that reason they perhaps tire fast, and they cannot be used for a prolonged amount of work. Now, what are the sources for energy? Well, before I go into that, One way to remember this is I always use chicken as an example. Now, when you think about chicken thighs and drumsticks, they're called red meat in a way because the meat is darker and, according to me, is better tasting. But that muscle, these muscles, are part of the legs. And how does the chicken use their legs? I mean, like anything else, to walk. But what I mean to say is that chickens usually constantly walk around. So they use them all the time. And for that reason, they're slow twitch muscles. What about the white meat of the chicken or the breast meat of the chicken? How is that used? I mean, chicken only use it when they need to fly. And how often do chickens fly? Well, not that often. They fly from time to time. And as you know, the white meat has poor capillary system, and like I said, it's fast twitch because it's only used sparingly, but the chicken uses it and then stops after a few seconds and rests. So you can use that example to connect the red muscles with the chicken legs and the white muscles with the chicken breast meat. Anyway, it's just an analogy that I use to help me remember. What are the source of energy for the muscle tissues or the myocytes? Well, that depends on what functions do they perform. I mean, someone might say, well, what function? They constrict and they relax. That's true. But it matters whether there is a heavy need of muscle constriction or the muscles are just at rest. If there is an outburst of heavy activity, the main source of energy is glucose that is degraded to lactose. And why is it lactose? Because if it's an outburst of activity, the supply of oxygen is not adequate. And as you know, to keep glycolysis going and to regenerate NAD+, so that NAD+ can be used by glycolysis, pyruvate is converted to lactate. Now, while that is good for the muscle because the muscle will continue to function, it can only be used to a point because lactate is an acid, that's lactic acid, and it changes the pH of the myocytes and its environment to more acidic, which is not good for the muscle tissue. So for that, the body has found a way to handle that fairly well. The generated lactate from the degradation of glucose or glycogen, then it gets excreted in the blood, and then it gets to the liver. The liver has a way of converting back lactate to glucose. And then that glucose now gets excreted into the blood for the tissues to use. And the cycle is closed. This cycle is known as the Cori cycle. It describes what happens with the lactate when muscles are required to do heavy work and prolonged amount of it, especially outbursts. Another source of energy for fast-acting muscles is phosphocreatine. Phosphocreatine is generated from creatine, which is phosphorylated by ATP. The phosphocreatine, though, is now available, and when there is a need for ATP, it acts as ATP and donates a phosphate to generate ATP. So it's like a phosphate donor. It's a quick phosphate donor. We generate phosphocreatine during times of plenty or recovery, like is shown here, but we use it during times of heavy activity to generate ATP. And what is the purpose of the ATP, if you remember from 481? Is to allow the myosin head and the actin filaments to connect and move with respect to each other so that the muscle constriction can actually happen. During rest or light activity, the muscles prefer to use fatty acids, ketone bodies, etc., etc. What it says here in blood glucose, but primarily fats and ketones. Now, jokingly, of course, I usually at this point recommend that if you need to lose weight, don't exercise. If you want to decrease your fatty content, just use light activity, such as, maybe vacuum clean or just move from a chair to a couch. That's a light activity, or keep at rest, and that's going to burn the fat. Now, that technically might be right, that the muscles then prefer the fatty acids, but of course we all know that usually people, when they're engaged in that kind of activity, their consumption of fats and other types of calorie is way more than the use of the energy as a result of the degradation of such fats and other calories taken in the body. So it doesn't work, as you already know. All right, there's another process that I want to describe, and that is the shivering. All of you have experienced it when you were cold. Shivering is designed to do basically incomplete or partial muscle constrictions, and they basically get constricted and relaxed very fast. And in that, ATP will be used, and when you use ATP, one of the products of that will be heat production. And when we break down ATP and produce heat, basically the body's way of keeping itself warm. All right, the last tissue that we're going to talk about is the neuronal tissue, or the neurons. They require a constant and steady supply of ATP because they need to keep the electrochemical membrane potential. As you know, if the membrane potential is inexistent or it cannot be controlled and regulated, neurons cannot perform their function. Everything, the signals are passed in the neurons by controlled change in the membrane potential. Their source of energy is mainly glucose and ketone bodies under starvation. All fatty acids and proteins are used indirectly. Someone might say, how? But I know you already know the answer. How can we use fatty acids indirectly? Well, remember, during starvation, fatty acids will be degraded to acetyl-CoA. Acetyl-CoA in large quantities, when it's in large quantities, will be used to generate ketone bodies, and then those ketone bodies are then used for energy. Same with proteins. During starvation especially, proteins will be degraded either to glucose, and the products will be used for generation of glucose or ketone bodies, which are the primary energy stores for the neurons. Now, why do they require a steady flow of oxygen and glucose? Well, the metabolic I shouldn't say the metabolic, but even though they do have a very active metabolism, it has been estimated that the neurons use about 20% of the oxygen we receive. Why is that? Well, because of this heavy need of ATP and the massive use of glucose and ketone bodies being burned for energy, and the needed ATP, the only way to generate it is if we have steady supply of oxygen. If you don't have steady supply of oxygen, you're not going to be able to generate sufficient amount of ATP that is needed for the function of the brain. And it says here that the neurons are sensitive to the blood level of glucose. Why is that? Because glucose has such great importance for the function of the neurons That's why they're so sensitive. As a matter of fact, I mean, you've heard about well, all kinds of regulatory functions. See, if you, for example, hypoglycemia, how does the body protect itself from hypoglycemia? By fainting. That's a defense mechanism. You know, we're usually afraid when somebody faints, but that's a defense mechanism. If the person doesn't faint, the tissues will not receive the necessary energy to supply the brain with the necessary energy so that the brain can perform its functions. So that's why all higher functions of the brain shut down, and the only thing supported are the basic core functions of the brain, such as keep breathing, keep your heart rate, temperature regulation, but thinking, vision, hearing, and higher processing, such, well, all of that I described, that's all shut down because it's not necessary. As long as the body can breathe and the blood circulates, you can keep the body alive. So just basic functions. All right, and with that, I will conclude this part of the presentation. I will continue with the rest our next time. Thank you for your time.